Hi, thanks. I guess I'll stand in between these. So today I'm going to talk to you a little bit about a voyage towards something that we hope gets us beyond the way that drug discovery is done today. And the key things that I'll focus on are how do we have models design what we try next and what we test next. And then I'm going to show you examples, and I'm going to talk about the work we're doing where things are very serial. We figure out what a target protein is that might, if perturbed or adjusted or bound, it might help cure or address or halt the prognosis of a disease, right? So we get a target, then we make a molecule, then we go to the clinic in all of the various ways we go to the clinic and manufacturing and all those things. So as we make a drug, it's very temporal, and a lot of that temporal ordering and that serial structure halts our ability or interferes with our ability to optimize this process. And so the two things that I kind of want to share, the two takeaway messages will be that with machine learning, we can do great many things that are in the generative space. We can make better molecules, and those molecules can do what they do with a lot fewer side effects, with better properties with respect to manufacturing and deliverability and accessibility. We can make better things. But today I also want to keep the undertone around machine learning because it can translate, because it has often the ability to see past missing data, because it is a great connector between things that aren't necessarily built to be connected. We also have an opportunity to use machine learning for process optimization, and whether that's in the clinic or whether that's in a vertically integrated drug discovery company, hopefully those are general messages that fall on receptive ears given the diversity of this audience. So I'll take the next slide. Do I advance with this? Oh yeah, perfect. All right, so before I start, thanks for the really great introduction. I was, it seems like yesterday I was a professor at NYU, but at this point we run, and we, myself, Kyung-Young Cho, who's still half-time at NYU, and Vlad Grigorovich, who's also one of our co-founders, we run the Machine Learning for Drug Discovery department at Genentech, which is sort of morphed into a function. And a function means the department is now multiple departments, but in my mind, it's still one thing aimed at not just coming up with better ways to design molecules, but also ways to foster basic research in that area and also integrate into the actual drug discovery pipeline. And so again, there'll be a tension in this talk where I say there shouldn't be a temporal ordering, we shouldn't do things serially, but every single example is still done in Roche, and therefore it goes through that serial process. I'll also say over and over again that there should just be atoms and interfaces, but I'm always going to show you examples that are either small or large molecule. And so I'm, you know, I guess I had to need some sort of penalty for being a hypocrite, like maybe no dinner or only one drink ticket tonight. But generally speaking, no matter what we do have to sort of deal with the momentum from the 100,000 employees at Roche that were there before us. And so we've set up our group so that we do have actually two units that attend to and provision AI representatives to all the active drug projects at Roche. And there are 218 of them. And that number, you know, if you care, could make you wake up in the middle of the night and not be able to fall asleep again. You know, it's a big number. But most of them are either large or small molecules. Because they're the same modalities, because these targets are grouped into families, that number is not quite as large as it sounds. And so we have about 20 people in each of these units who collectively try to make sure that each of these projects has the right data infrastructure, the right machine learning infrastructure, so that the designs made are primarily and essentially with near 100% completeness pass through machine learning models before they are tested. So even though we have lots of biology that makes these projects different in their own way, we still make sure that every molecule is at least put through the model and we know where it is in the space of the model compared to things we've tried before. And I'll talk about examples from these two things, but I'll mostly talk about examples from the small molecule side, which is a little bit newer for us. The core of Prescient was the Frontier Research Group, which is run by Kyung-Young Cho, and I think I'll show a good amount of work from this group And I'll point to a handful of papers that are in this active learning meets machine learning space. The machine learning engineering group, you could imagine a lot of what we need to do is we need to take our scientists who are working on molecules and core machine learning, and we have to find ways to give them infrastructure so that they don't have to think about the fact that they're scaling this up to a very large number of people and projects. These folks very often, kind of get so involved in these projects that we have to just kind of let them rotate to these groups. People get excited when they think they're close to having a therapy that could actually make a patient's life better. And so the attrition from these groups is nuts. Like every 2 years, about half of these groups decide they want to come over here and just be part of this team or that team or the colorectal cancer team or the Alzheimer's team or whatever. And so in spite of our best efforts, no one listens to me, and we're constantly migrating people here. But we do have an infrastructure here, and I'll show you some of our agentic frameworks and things like that. And even though there's a lot of day-to-day here that's not that exciting, data hygiene, getting data ready for machine learning, still something we got to talk about. And I think there's also a lot of cutting-edge research that comes out of the engineering group in the space of LLMs and agents. So that's exciting enough that we can keep a few people in this group. And then we do have our sort of foundation models group. And here, this is the group that tries to remind everyone that each project is not unique. We shouldn't have to start everything anew, and that we do need to make sure that the large foundation models that we use, and also the large foundation models that are provisioned by our partners or by the open access, open source community, that those foundation models are used so that we're not retraining every time, so that we are maximizing the degree to which we can transfer knowledge between projects. So that's the setup for our unit. And I go through it just because I think the sort of balance between praxis, connection to the actual medicines we're developing, and the core research, this is a balance that we discuss quite a bit, that we try to get right. And like anything in the real world, it involves the difficulty of recruiting people, the advancement of projects, the excitement of getting close to the finish line. And so we do our best, but it's at best a dynamic equilibrium. And so what I'm showing you here is our ideal envisionment of what this would look like. All right, so now, when we think about making a drug or medicine, and Genentech corporate communications wants us to always say medicine and never say drug. So if I say drug, just hear medicine, bear with me. Okay. So a lot of what I'll talk about today too, I want to just quickly give a shout out to Sarah Mustafavi. And so she is the new vice president of computational biology and translation. We work together very closely because I can't do anything unless I know what targets I'm going after. And so her shop is working very closely with research biology to provision machine learning that helps prioritize targets and connect that to tractability, which is kind of halfway between us, and safety, which also involves both molecular and biology aspects. And so as we move forward, what we really want to do, a key thing that we want to do is first of all, make this system integrated so that keeping track of the drug discovery process happens with machine learning assistants that have access to structured data that goes from pathway to target, goes to molecule, and then comes back again to the biology. Because once you have a molecule that works, then you need biomarkers and other things to run a clinical trial and anything approaching the modern sense. So what helps you figure out the target leads to, in the ideal system, leads to ensembles of targets that are held in substantially more sophisticated data structures than the serial process needed, where we have targets. As we're prioritizing targets in the early research part of things, we're also looking to see if they're druggable by queuing up these simulations. So this crazy thing here where we have orchestration, interface, and our data system in the middle, we have the biology and the drug discovery talking to each other. In the beginning, the tangible system involves anytime something's being looked at in research biology, queuing up automatically the generative AI to design drugs, given that set of ranked targets. In the beginning, you don't know what modality is the right modality for perturbation, and so you try antibodies, you try small molecules, you try peptides. That can then perturb the ranks here, and if you perturb the ranks here, that's not just a linear process, that's not just an additive or multiplicative process. This is a pretty dynamic system. And then these different color arrows here, it's important that they're differently colored. This indicates that people are messing around with stuff. Somewhere in the middle, we still do have people like Yan Wu, who is one of my key partners. She's the vice president in charge of antibody engineering, and she's literally made 7 drugs that have saved huge numbers of people, and she did it essentially by hand in the mid-90s onwards. And so she knows a thing or two, and having interfaces that she can play around with and her people can play around with is extremely important. But again, it's unpredictable, and that's why we have these multiple colors that I really thought about altogether too long. Today I'm gonna dig into, that's only 5 minutes of hot air, so you guys are super lucky. So 6, sorry, I'm a minute over on the hot air. But I'm gonna focus today on these 2 things. Mostly I'm going to focus on what we do with Lab in the Loop, which is really Lab in the Loop for drug design. I'm going to focus less on Sarah's part, partly because that's her talk and not my talk, but also partly because I think there's plenty to get into in a half an hour in the molecule design part of this. If there's time, and there probably won't be, because there's already been 2 minutes too much of hot air, I'll get into agentic frameworks and some simple examples of how those can be useful in this context. Before I get into that, I'll also just say that there's 2 things that keep getting convolved here. There's the actual theory about how you properly integrate these things so that you have target rank, pathway rank, the target choice. You've got the molecule, and then when you start the clinical trial, you have biomarker information. Having all of those data structures present at each point throughout the system, just doing that, it reduces the activation barrier to hoping to make this process less serial. And so that's a key thing here. The data issues involved in this are extremely challenging, and so we've got great partners in Anna Craig and Ryan Copping, who are both Roche veterans. They've been at Roche for 20 years, and they're helping us to wrangle the 125 different systems that span 125 years. And those numbers, unfortunately, are not exaggerated. Other things in this talk will be exaggerated, but not that. All right, so if we just want to use machine learning to iterate, optimize seeds, drugs, or to hit targets where we already have some biology that lays out what the pocket is and what the epitope is, what's the key function that we want to perturb, we still have a long road between there and a molecule that can be put into a patient without eliciting immune response, that can be put into a pen so that we can mail that pen to rural locations that don't have refrigeration, right? Some of the things that we do for accessibility, high concentration stability, they end up being pretty advanced by the time they get into the ways that people want to administer drugs beyond infusion. And so this Lab in the Loop is the key to everything. And here we have active learning and drug discovery integration. And so the part of this is praxis, part of this is theory. The drug discovery part, that integration, it means that every single instrument in the path of designing a drug, we need automated data capture that goes back into our systems. All right, so I'm saying that not because we're going to have great conversations about that over dinner, but just because that's difficult, and we're getting there at Roche, which has got me very excited. But it's kind of like very excited in a very down-to-earth way. The active learning part we'll go into a little bit, and I'll show you what some of the key challenges are there. But here, the key thing is that you want to make sure that what you're testing next is diverse. It's driven by the model, so that if it works or it doesn't work, it's going to always improve the model. Balancing the exploitation and the model improvement aspects of this, we'll focus on that a little bit. The other challenge is that drug discovery real parameters are very diverse. Some things involve interaction with the whole human animal, the patient. Other things involve molecules and even the quantum mechanics of bond strain. And so you really span a lot of scales here. And lucky for us, machine learning can be formulated in a very flexible way there. The generative AI for molecular design, here I'll show you just a little bit about how we do that in a cross-modality way. And so we want to make sure that this is very tuned to a project and that the rest of these things are actually single systems that we can maintain for all the different ways you might make a drug. Agents and orchestration, for us, this is a real lifesaver, again, because these systems are flexible enough that you could hope to make a small number of systems that scale to a big org. And if we had to, for each instrument, for each assay, for each project, design interfaces or make things accessible otherwise, we'd either need to teach everyone at Roche Python, or we would need to, which I guess we could do with vibe coding and things, or we would need a system that you could talk to. And at a place like Roche, just finding the data, even with structured systems, with a system that you can talk to and iterate with that also knows where tools are and how to connect data to tools, this is something that's a current reality and has been a really amazing to see this in operation this summer. And then the fact that machine learning is really also a way to just motivate process optimization across big organizations. I think this is something that we'll skip for today. All right, so lab in the loop for drug discovery. This is what we'll get into today. And really, in the beginning, even though we have ways of directly designing molecules, right? We also do have very, very straightforward ways of making antibodies. With mice and rats, and at Genentech we have mice and rats who have been genetically modified so the key parts of their immune system are human. So you immunize with a protein of interest, an oncogen, a surface marker, a receptor that you want to antagonize or agonize. That mouse then generates a human antibody. And so those things, there's a lot of sunk costs, but if we want to operate that, it's very economical to pull out thousands of binders for a small amount of money in a small amount of time with a small number of people. And so, and we also have DNA display technologies, ribosome and yeast display. And so one thing that when we landed at Roche we realized was that even though we know how to de novo design molecules, we also can get them very quickly in these very economical processes. And so we usually start off with a mixture of antibodies that are some coming from de novo design, some coming from the GenN mouse, and some coming from yeast display. And so it's really exciting that when we really care about something, it's an exciting target, it's been prioritized, we want to find a tool molecule for it right away. We usually have molecules from 3 really distinct different processes right away. And that's exciting because there are lots of biases in each of those methods, right? If you do de novo design, your structure model for the target that you're trying to target is a huge bias. Nobody's talking straight about that. Even though we can get these things to work, we know that structures aren't static, and there's a lot of thinking and modeling that has to happen before you know what structure or what form you want to target. The GenN mouse has a limited human repertoire, so that's a huge bias. Our yeast display libraries are biased in so many different ways that take an hour to describe, even though they work. So they all work, but we get very punctate clusters. We put those in. Once we get that, we go through a system that, given the models and given the data we have together, based on the models and the data, tries to produce a Pareto front that tolerates the fact that there's a lot of missing data, pushes that out and tries to produce, or does produce, a set of designs. These designs are both libraries that test 10 to the 9th, but we're modulating the distributions. There are also plates of designs, typically somewhere between 100 and 1,000 designs that we test per round. We put that back in. Natasha updates these. The models update. We turn this crank one more time. Now, the problems are many, but one thing that is, an argument for active learning, it's not an argument, it's the argument and the winning part of the argument. You can't use machine learning, you can't use high throughput biology, you can't use any of these things in a design prototype cycle, which is the only way you design things, if you don't have some way post-inference of selecting a really small subset. And whether you're doing that in your generative process and the scoring function is just really narrowing things down, or you're doing that really with your eyes wide open with an active learning like framework, you got to do that. And so what we tend to do is we don't put all the pressure on scoring. We know that we have a fairly large machinery to test things, and so we use things like property DAG and Bowtide to select the correct subset that balances exploitation and model improvement. And Jiwan Park who is one of the earliest members of Prescient, Natasha Tagasowska. I'll show you a couple of their papers. But if you look for this property DAG, multi-objective, blah, 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 NeurIPS paper, workshop paper, this will lead you to the rabbit hole that has all of these papers in it. OK, and so property DAG deals with the fact that these are really complex, interconnected, nested properties. And bow-tied is the sort of neural empirical Bayes framework that goes around that to select the right subset. But here's the principle, right? So again, not everybody has been fighting this out for the whole time. And so here's the basic principle. When we want to use machine learning, and we have 16 different properties on the road between a tool molecule and a medicine, and if, here I've got affinity to the target and the expression yield, which is, actually pretty important. And also, expression yield in different systems has a few mysterious elements to it. The DNA, the this, the that. It's not just stability. The sort of model-driven experimental design MO that's, about 110 years old is that we optimize the hypervolume that we explore with any predictors, with any experiments we do, with anything, right? And if you actually look back to early, Pearson's work where he says block random is the best way to do things, and that was the MO for the FDA for about 50 years. Block random's not too bad. If you do block random in a high enough dimensional space, you're getting pretty close to if you took the if you took the the determinant of the inner product of the data points, and you tried to maximize that, that's how you maximize the volume of a big high dimensional space. If you did that a whole bunch, you wouldn't really beat block random by very much. Okay, but with a lot of biases and a lot of structure to your data, you should pay attention to this. And so the idea is you have to push this volume out. And the terminology for people who are not in this field is that this blue point, for example, would be a new point. These blue and orange points, they dominate these other points because when you add these points, the hypervolume takes these points that used to be on the frontier and it puts them on the interior. And so this is sort of optimization speak for picking a good point. You pick the points that dominate. But what is the actual reason why this is a disaster, very complex thing? It's because if a protein doesn't express, you can't tell me how much it expresses because it died for some reason. The DNA had some cleavable element, it was toxic, who knows. If it doesn't bind, I can't get anyone at the company to care about it anymore. If it's looking like it's not human, then it's also not going to be a drug, and so we're not going to see how long it lasts in the bloodstream. We're not going to see whether or not we can concentrate it to 200 milligrams per milliliter. And so there's a huge amount of hierarchy in these, and instead of going through all of that, I'll just say that is solving that problem is what we talk about in this property DAG paper. The fact that if you know the relationships, you know the order, and you know the tree of dependencies for variables, then you can actually get to a framework that's simple, like these Pareto frontier models, actually pretty quickly. And the trick that Jiwon and Natasha use is to kind of use Vine copulas and work in the interior space, pairs of data points. And this actually allows them to do a lot of things in a mathematically efficient way that they wouldn't have been able to do otherwise. So this is old work, but I like to highlight it. It was essentially what powered our first big push was Antibody Lab in the Loop. This is, again, a paper by Nathan Frey and his 52 closest friends, and then all the old farts who pay the bills at the end. But basically what we found is that if we looked at multiple properties here, and these are properties like predicted immunogenicity, viscosity, et cetera, what he found was that we actually could get to something where when we thought we would, by selecting these designs, push the Pareto frontier, we actually did. So the first design was okay. We had binders. We had things that are at the level or better than most of the binders you see in most of the papers that come out of biotech startups, which is what we were in the beginning. So here we are. We went through multiple rounds, and we pushed the Pareto frontier with each round in different ways. So expression for round 2, we got back to some nice balance. And then expression for round 4 doesn't dominate in every aspect, but the key parameter we were trying to optimize, PKD, dominates. And this doesn't look Super impressive compared to a lot of the pictures you see in theory papers, but this is actually with proper therapeutic antibodies that at the time were active drug projects. And the reason we were able to publish them and talk about them is for whatever reason they were attrited projects. So we either partnered with another company that had a different molecule that worked better for that disease, or there was something in the biology that didn't work out. So these are molecules that could have been, should have been, but the biology changed on us. Again, I keep talking about process optimization, which is a weird thing to talk about, but I want to just really quickly show. Actually, let me use the slide the way it's meant to be used. You know, if we have de novo ability to design antibodies, which at this point is now a commodity technology in many ways, it's arguable, but I think it is, then we can paint an epitope and bind the antibody. And we skip the need for animals, immunization, all sorts of things. And so most people think that that means that we can just make binders, but we can also take existing binders. If you can make things from scratch, you can also take big sets of prior data, 100 years of data, naive repertoires from people, all kinds of things, and you can figure out what's happening in a lot of settings in terms of where you're binding, and ultimately get to a cleaner picture of structure function. Because when we have antibody drugs, they don't just bind and turn things off. They bring things together, they push things apart, they block one part of an allosteric function and not the other. So the epitope is super important, and that structure-function relationship is never simple. We can also start to think of complex formats. So typically when we get involved in projects these days at Roche, we have a targeting arm or a T-cell recruiting arm. Some of the arms sense pH or ATP, the tumor environment, others don't. There's other sort of environment sensors for other indications like inflammation. We also need to essentially figure out some of the ways in which we can make this faster. So we can actually start to think about deploying these molecules. If we discover some biology we're interested in, we can start to ask the biologists to incorporate tool molecules and instead of genetics much earlier. And then lastly, like I've already said, I suppose, we can be much more, much cleaner about structure function hypotheses if we have a model of the structure at every step of the way. And so I've talked to you about optimization, lab in the loop, active learning. I think these methods, they go beyond just optimizing expression, immunogenicity, binding, but they also let us think about new ways to do this that are beyond just optimizing the existing processes. So that'll be interesting. On the small molecule front, we have a similar recipe overall, an active learning loop that we would like to just call an active learning loop, but our bosses want something flashier, and so it's lab in the loop for everything. So we, again, corporate communications intrudes. But we do have active learning, and again, here, there's a lot less missing data. You either can't make a small molecule at the beginning, or you can, and if you can, they tend to test everything. And so we find that the process that we're dealing with, the giant company of what, that's spending about $10 billion a year testing all these predictions, what they test and what they don't test, it's really sparse, but it's all at the root. That said, Natasha and Jiwon's magic property, property DAG plus bow tie still works. There's a lot more physics here because since you're synthesizing these molecules, they're conformations, ways in which you can modify them, it does involve a fair bit more physics meets machine learning. Instead of de novo, they like to call it hit finding. So there's different language, but we do have a lot of work here to generate designs from scratch. And there is plenty of conformational sampling, but it's less on the protein side here and more on the internal degrees of freedom side. And one of the things, though, that we really want to do is, given how similar the recipe ends up being, we want to think through ways we can push beyond being stuck in single modalities. And so I've got only about 3 minutes, but I'll just give you the key pieces. So Pedro Pinheiro and Matthias Kirchmeier, these are the two names that will lead you to a lot of the papers that describe funcbind, voxbind, and all these various things. But basically the idea is Pedro and Mathieu came to us from Vision, and they said something that actually turned out over the last year to be true, that you don't like voxels and you don't like Vision stuff because when molecules rotate, it shouldn't matter. But rotation and translation is just a quaternion. So you're telling me that this thing with 1 million parameters can't learn 4x4 numbers. So they said, we believe that if we use voxel representations of protein surfaces, all the equivariance and rotation invariance, all that stuff will just be in the first layer of the neural network, and it won't matter. And it turned out they were right. And then they figured out how to better condition on dynamic pockets. Then we started doing things with antibodies, and they ran out of memory on even the biggest instances. So they figured out how to reframe this with neural fields as a sort of latent encapsulation of the voxel. This is a little bit just machinery. It's not really deep theory, but that then eventually led to FuncBind, which is a way of conditioning on a protein pocket or an ensemble of dynamic states of a protein pocket to generate molecules into that pocket. And so this is kind of what it looks like. You take a piece of your pocket. You've got some things you're generating, some things that you're keeping static. And we can set this up so that we actually can do it in the space of just atoms, right? And so we have a voxel representation, which eventually gets converted into a neural field, essentially a latent, very efficient encapsulation of this voxel. Eventually, we're generating atom clouds, and then we're only turning them into antibodies, peptides, or small molecules at the end. Now, you still do need some variables that tell you in advance what kind of modality you're trying to make, and that's because we have big foundational models that tell us what is a valid small molecule, what is a valid peptide, what is a valid antibody, and that has to do with the fact that antibodies have germlines. You can't make every small molecule. Peptides have libraries of amino acids that are cheaper or more expensive, and so we need to condition on what we make, but we do that just with a single variable that indicates the modality. Whoops, that's the, and so we encode, we build out clouds of atoms, and then we decode conditioned on the expected modality. And the other part of this that's important is that the framework, I don't think these are particularly useful slides. Let me actually go to this one. The other part of this that's particularly important is that we use Saeed Saremi, who some of you might know from the early days of neural empirical Bayes. And the early days of machine learning means like 12 days ago, 8, this ancient seminal paper from a whole year ago. Anyways, Saeed Saremi in about 2018 started working on neural empirical Bayes, worked with a whole bunch of different people to come up with a bunch of some things there. And at present, he's developed that into what he calls the walk-jump sampler. And so this is an idea that diffusion is great, but it's got all these parameters, and you're slowly boiling and cooling and cooking, and instead of having 20 to 1,000 hyperparameters for the noising and denoising process, walk-jump has just a single parameter. And so when we are exploring the noised space and we jump to the denoised space, we do that in a single step with a single parameter that isn't a hyperparameter but is controlled by the variance in the input training data. And so the idea is we move around, conditioning, generating into this pocket. We do it with Saeed's sampler. And again, start here if you want to actually understand it. We jump, we decode. We can score in either of these two spaces, the denoised or the decoding step. So it's a lot like a diffusion process, but it is a walk-jump process. And if I said it was a lot like a diffusion process without first introducing Saeed's work, he would find me and beat me up. So I'm not going to do that. These movies, these are the last frame of movies where we generate antibody loops. We also can, with the same code, with some modulation of the input target, obviously, we can generate small molecules, in this case into a nuclear hormone receptor and another gene that I am honor-bound not to name. But these are essentially active projects, real projects. And we can generate macrocyclic peptides, which are, right now through obesity and cardiovascular, they're having a resurgence of importance as a modality. And I like this because I can take a lot more people, get them to really feel like they have friends when they're talking about this, not have to have different isolated groups working on different modalities. But I think the other thing that's exciting about this is it brings back the need for a lot of physics-based scoring and a lot of real thinking about what is the right way to inhibit these interfaces that's broken free from some of the bias from the three major ways that we make drugs, right? We've been having a lot of fun playing around with this, seeing when it crosses over between one modality and another, and this is kind of an interesting example where we had a known binder. We generated into that pocket, and we found cyclic peptides that actually ended up docking in a similar way to the known binder without having that information prior. They also imagine new side chains that, according to the chemists, are synthesizable. They're compatible with this sort of macrocyclic format, but they're not side chains they had tried before. And so it's not just extrapolating within the set of side chains it's seen before. It's having fun and imagining new chemistry that the chemists aren't disgusted by. Early rounds of generative AI had a lot of chemists laughing about how this would explode or this would oxidize, anyways. All right, so I think I'm going to hand off and just say that, in general, our multimodal approach to this is working. I talked to you a little bit about the overall framework, and so we dove down into FuncBind, which is a way to make those binders. All of that would still have to fit into this overall framework, and I'm going to skip. We didn't get to talk about agents, which is good because I don't think the lawyers really like us talking about agents. And I just want to thank what is really one of the best teams that I've ever worked with. Kyung-Young Cho, myself, Franziska Seeger. We've had a lot of support from Aviv Regev and the leadership at Genentech to build this team. Here we are at the main campus on Genentech. And I think I'll stop there and hand off to our next speaker. And thanks, everyone.